in the cell structure and functions, we are studying about the different organelles present in the cell. So far we have studied about the cell membrane, which is explained by Singer and Nicholson. What they explain the fluid mosaic model, the cell wall, its chemical composition, and various organelles like endoplasmic reticulum, Golgi complex, vacuoles, and lysosomes, and then non-living, non-membranous organelles like ribosomes, then centrosome, then structure of cilia and flagella. So these are the things what we have studied. Other than this, the important organelles what we have studied is the mitochondria and the plastids. So these organelles have their own specific function which is essential for the cell activity. So for example, mitochondria, it helps in the production of energy during a process called as cellular respiration, and also stores energy in the form of ATP. So that is why mitochondria, we call it as the powerhouses of the cell. And similarly plastids. So based on the type of the pigments, we are having the chloroplasts, chromoplasts, and leucoplasts. Chloroplasts contain chlorophyll and carotenoids. Chromoplasts contain xanthophyll and carotene. Leucoplasts contain no pigments, and they are usually storage and function. So when we study these, they are having their own function. The next about cilia and flagella also we have studied. It is made up of microtubules which are arranged in doublets of 9 pairs, and similarly the centrosome consists of a pair of cylindrical rod-like hollow structures called as centrioles. So these centrioles also are made up of triplets. 9 triplets are there which are made up of microtubules, and these microtubules are the main component of the centrioles. Now the region surrounding the centriole, it is a dark amorphous material which is called as the centrosphere. So that is how this centrosome, which is present usually near the nucleus, the centrioles of the centrosome, they arrange at the polar ends during cell division and produce the spindle fibers. So that is about just a brief explanation about the certain parts of the cell organelles. Now another important organelle in the cell is the nucleus. So the nucleus is the important component of the cell. Now suppose this is a cell, so you will find the nucleus in the center usually. And coming to the structure of the nucleus. So a nucleus is made up of a double membraned structure. Now suppose this is a nucleus, this is the membrane, and this is another membrane. So it is having on outer and inner membrane, but together these two membranes are called as the nuclear envelope. So the nuclear envelope is the membrane which is present. So this nuclear envelope, so this is the pore. So these two nuclear membranes. So this is the nuclear pore. The nuclear membrane is having pores like this, and so this is the nuclear envelope or nuclear membrane, and this is the nuclear pore. So the nuclear pore communicates with the outer cytoplasm. So this nuclear pore is communicating with the cytoplasm, and inside this there is a fluid which is called as nucleoplasm. So nucleoplasm is also called as karyolymph. So that is the fluid present which is almost similar to the cytoplasm. So karyolymph or the nucleoplasm is the fluid present inside the nucleus. So the nucleoplasm is communicating with the cytoplasm through this nucleopore. So the nucleopores, they communicate between the cytoplasmic region and the internal part of the nucleus, so where the nucleoplasm is present. So in other words, the nucleoplasm and the cytoplasm are communicating each other. So they are in contact with each other through the nuclear pore. Now inside the nucleus, we shall see what is present. So inside the nucleus, there are fine thread-like structures. So these fine filamentous structures which appear like a network. So this is the network, and these are called as chromatin threads. So chromatin fibers or chromatin threads. So the chromatin threads are transparent. So they are fine transparent thread-like structures or fibers which are freely present in the nucleoplasm of the nucleus, and they are appearing like a network. So sometimes it is referred as the chromatin network. So the chromatin network means it consists of these chromatin fibers, and this chromatin fibers which are present, they contain or they are made up of DNA, they are made up of proteins, and those proteins are histone proteins and also non-histone proteins. So the chromatin is made up of DNA and proteins, in other words. So this chromatin which is made up of DNA and proteins, so the proteins are histones and non-histones. That is the composition of chromatin. In addition to this, inside the nucleus there is a spherical body, a dark spherical body. So this spherical body is called as nucleolus. So this dark body which is non-membranous, again it is not made up of membrane, it is a also certain amorphous material. So this nucleolus is a spherical body which is dark in color, and this nucleolus contains rRNA and the certain ribosomes. So this ribosomal RNA which is associated with the ribosomes is present in the nucleolus. That is actually the content of the nucleoplasm. So this nucleus is usually spherical in shape. Now in a cell, usually a single nucleus is present, and this nucleus is usually located in the center of the cell. Sometimes it may be towards the periphery or away from the center of the cell, as in case of some plant cells, for example in parenchyma. Due to the presence of the vacuole, the reason is as the vacuole occupies most of the cell area, so the nucleus is shifted or it is displaced towards the peripheral part of the cell, and there we find the nucleus in peripheral in position. And in other cells, for example in animal cells if you take, the nucleus is usually located in the center, and sometimes the nucleus is also located towards one end of the cell, as in case of the columnar epithelium which we have studied in the animal tissues. So there the cells which are elongated, narrow at the basal end, broader at the free end, so the nucleus is present at the narrow basal part of the cell. So that is where the nucleus is present towards one end of the cell, that is the position of the nucleus. And rarely the cell may contain two nuclei. The cells are binucleate. For example, some of the liver cells, they contain two nuclei, binucleate. And even in other examples, for example in paramecium, the cell is binucleate, and again the two nuclei may be same uniform size, or they may be there may be variation in the size. In paramecium, one nucleus is larger and another is smaller, so that is why it is called as the macronucleus and micronucleus. Usually the macronucleus is kidney shape and the micronucleus is small and spherical in shape. That is the shape of the nucleus. And sometimes the cells may be multinucleate also. In case of muscle cells, we have studied in the animal tissues, the skeletal muscles which are cylindrical shaped and unbranched. So each muscle cell or each muscle fiber contains numerous nuclei which are usually located towards the periphery of the cell. So peripheral position of the cell, that is the what is the number of the nuclei in each cell. So in most cases the cell contains single nucleus. Rarely there are two nuclei, and also there are many nuclei, multinucleate in condition. And the nucleus function, as there is DNA in the nucleus, the chromatin is made up of DNA. So hence the nucleus is the controlling center of the cell activities. So all the activities in the cell, whatever may be the activities, so these activities, they are controlled by the nucleus. That is because of the DNA present. So the DNA is the genetic material which is present in the chromatin. So the DNA is the master molecule which controls all the functions. So it controls all the expressions of the characters. So the expressions of the characters, they are all controlled by the DNA. So it is due to the activity of the DNA that where the characters are expressed. So hence whatever activity is to be done by the, or is to be performed by the cell, that activity is controlled and it is expressed by the DNA activity. So that is why the nucleus is called as the center of the cell activities. It is the controlling center of the cell activities. That is about the structure of the nucleus. And the shape of the nucleus, again, the number of nuclei, it is 1, 2 or many. The shape is usually spherical. Normally the nucleus is spherical in shape, ball like, or sometimes it may be oval or sometimes elliptical. Rarely it is somewhat horseshoe shaped, means somewhat curved like. In case of some WBC we come across, and sometimes the nucleus is lobed, means it appears to be many. Now suppose this is the cell, and this nucleus is split into small lobes like. So this is one nucleus actually. The nucleus are linked by chromatin fibers. So this is one part of the nucleus. So lobed nucleus it is called as. So in again WBC, some of the WBC, the nucleus is split into lobes. Now these lobes are not free. The lobes are connected to each other, so that all together forms a single nucleus. So all together form a single nucleus in the cell. That is how the nucleus may be lobed nucleus, it may be horseshoe shaped, and already mentioned in the paramecium, the large nucleus is kidney shaped, and the nucleus is, in most cases, it is spherical in shape. So that is how the nucleus may be having different shapes in the cell. Now next about the chromosome, study of the chromosome. Now what is chromosome and what is chromatin? Now when the cell divides, especially during the mitosis or meiosis, so whenever the cell division is initiated or it prepares for cell division, these chromatin they are transformed into chromosomes. Now chromosomes are seen only during the cell division and they are very clear during the metaphase of the cell division. Now this chromatin, now suppose this is a chromatin. So the chromatin is a fine transparent fiber-like structure or a thread-like structure which is coiled. It is not straight like this, but it is folded in various forms. So this chromatin is present in the nucleoplasm, which contains DNA and proteins, which may be histones and non-histones. Now this chromatin fiber at the time of cell division, so these chromatin fibers cannot be seen clearly under microscope because they are very fine and they are transparent in appearance. So because of their transparency, so they are not distinct in the cell. Now this chromatin in the nucleus, it will condense, it will get shortened and form a cylindrical rod-like structure like this. So it is just like the condensation of a rubber fiber. Now if you take a rubber and you can stretch that rubber and you can make the rubber contract. So assume that the rubber is shortened or contracted, condensed. So when that rubber is condensed, it is shortened, it becomes thick and short. So the chromatin fiber, which is having certain length, it gets condensed, and as a result of condensation, the length will decrease and it will the chromatin becomes thick, rod-like or somewhat cylindrical in shape. So this rod-like structure which is formed is called as chromosome. So this is called as chromosome. So chromosomes are rod-like structures. So they are structures which are seen only at the time of cell division. So the chromatin condenses into chromosome at the time of cell division. So the condensation will start at the beginning of the cell division, either mitosis or meiosis. So when the condensation starts, so the condensation goes on, and as the cell starts dividing. So when the cell starts dividing, so we come across prophase, metaphase, anaphase and telophase. So during prophase, the fibers, the chromatin fibers are on the way of condensation, and during metaphase they are maximum condensed. So during the metaphase stage of the cell division, so the chromatin are condensed maximum or to large extent, and they are very clear during metaphase. So whenever chromosomes are to be studied, whenever the structure of chromosome or any characteristics of the chromosomes are to be observed, so it is better, or it is usually observed during the metaphase of the cell division stage. So that is what is chromosome and chromatin. So the chromatin itself, they get transformed into chromosome. The chromatin gets condensed, they get shortened and become thick, rod-like, and that is chromosomes. So how many fibers are there? so many chromosomes are formed in the cell. So this chromatin fibers, so this is not a single fiber in a nucleus. So there are many such chromatin fibers which are folded and they are almost appearing like a network, and these chromatin fibers individually they get condensed and we can see the chromosomes at the time of metaphase during the cell division. So the number of chromatin is equal to the number of chromosomes. So how many chromatin fibers are there? So many chromosomes are formed. So the number of the chromosomes is constant for a species. So for example, in human being there are 46 chromosomes. So that means there are 23 pairs. So usually the chromosome number is in even number because the chromosome are in pairs. Now this pairing is very clear and distinct during meiosis, and again during mitosis cell division we don't come across pairing. So totally there are 23 pairs of chromosomes in the human being or 46 chromosomes in the human being. So any human being, so any means whether it may be a male or a female, or whether it may be a newly born child, or it may be a middle-aged person, youth, or even an old-age person. So whatever may be, irrespective of age, irrespective of the sex, the chromosome number is constant. So even in the human race also the chromosome number is constant. So whether the person is belonging to Chinese or a European race or any other country, there the chromosomes are constant. So the chromosomes are constant for a species. There are 46 in human being and there are 16 chromosomes in onion, 12 chromosomes in pea plant, and there are 44 chromosomes in chimpanzee, monkeys, which are the close relative of human being, and there are 70 chromosomes, 35 pairs in a dog, and there is a fish which is called as goldfish. In that we come across 100 chromosomes, 50 pairs. So like that the chromosome number varies, and there are even nearly 1000 or more number of chromosomes in some organisms. For example, in one pteridophyta plant, a fern, so in that we come across more than 1000 chromosomes, about 1370 or something like that. So like that the chromosome number are varying from species to species. The chromosome number is constant for a species. So in insects there are 8 chromosomes, that means 4 pairs, and like that different species are having a different number of chromosomes. Now when we study the structure of a chromosome, what it deals with. Now suppose when we take the structure of the chromosome, so the chromosome is made up of fiber like this. Chromatin fiber is coiled. So the chromatin which is stretched here, it condenses. During that the coiling of the chromatin takes place. So this is the chromatin, how it is coiled. So in some regions the coiling is not present. So this is the chromatin which we can see. So this chromatin is coiled in various manner, and that is why it gets shortened. So when this chromatin is stretched, it becomes straight. So this condensation is because of the coiling, tight coiling or very closed coiling of the chromatin. And here the coiling is absent in some regions. So this region where coiling is absent, is called as centromere, or sometimes it is called as primary constriction. So this centromere or primary constriction is the region on the chromosome where coiling is absent. Here the chromatin becomes straight and does not coil in this region. So this colorless region or a somewhat transparent region, that is called as the centromere or primary constriction. Now the tip of the chromosome, this is called as a telomere. This is the telomere. This is the telomere, then that is the telomere. And then in this centromere, here there is a pair of cap-like structures. So you find two cap-like structures which are present in the centromere region. So these cap-like structures which are present, they are called as kinetochores. So the kinetochores which are in pair, they are somewhat C-shaped or somewhat half-moon shaped. So they are in pair present in the centromere. So the kinetochores are having microtubules on its surface. Actually, when we go into more structure of that, so the kinetochores are having microtubules, and these kinetochores help in the formation of the spindle fibers or the mitotic apparatus. That is the function of the centromere or the kinetochores present in the centromere. And the region of the chromosome on the either side of the centromere. So this is the centromere, this is one side of the chromosome, this is another side of the chromosome. So this region of the chromosome is called as chromatid, or sometimes it is called as arm. So this is also one chromatid and that is also one chromatid. So in other words, the centromere in the chromosome divides the chromosome into a pair of chromatids. So the centromere may be present anywhere along the length of the chromosome. So usually it is present in the center or away from the center. Now this from this end to this end, the centromere is present anywhere. So the centromere or the primary constriction divides the chromosome into a pair of daughter chromatids. So this is one chromatid and that is another chromatid, which is also called as arm. So the chromatids or arms, they are the part of the chromosome that is on the either side of the centromere or the primary constriction. So the free end of the chromosome is called as telomere. So the telomere is the end of the chromosome, which is very important. It will prevent the attachment of the chromosomes in the cell. Now suppose this is one chromosome and this is another chromosome. This is another chromosome. Like this, the chromosomes are present in the nucleus of a cell. So in the same way, there are 46 chromosomes in human being, the cell of a human. Now these chromosomes which are present, now sometimes when they come in contact like this, the telomere and the telomere of another chromosome, that telomere will prevent the attachment of one chromosome to another chromosome. Now suppose if this chromosome and this chromosome gets attached in case, so this will become considered as a single chromosome. So when these two chromosomes attach, so then this will be considered as a single chromosome, though the chromosome may be very long because of the attachment of two chromosomes, but it is functioning as a single chromosome in case if it gets attached. So when the two chromosomes get attached, the number will get decreased. So in human being, if you take example, there are 46 chromosomes. Among the 46 chromosomes, when two chromosomes get attached, the number will become 45 chromosomes. So like that the chromosome number will change. So if there is a variation in the chromosome number, it will lead to some disorders. So the chromosome number, though they are 46, but they should be freely present and they should function independently. If they get attached among themselves, so then the say two chromosomes function like a single chromosome, and it affects the total chromosome number of that cell and result in certain chromosomal disorders which are caused due to the change in the chromosome number. That is why the telomere region is very important. So the telomere will not allow, it will prevent the attachment of the chromosome, that end of the chromosome will prevent the attachment to another chromosome, and thus the chromosomes are functioning separately and freely. Now that is about the chromosome. Now just some more important more extra information about chromosome. Now this is the chromosome. Now the length of a chromosome may change again. Now in a single cell itself, so though there are many chromosomes, all the chromosomes will not be equal. And here whenever two chromosomes are similar, they resemble in all characteristics, in size, shape and everything, and other all characteristics. So such chromosomes are called as homologous chromosomes. So here homologous chromosomes will pair among themselves. So the pairing of the chromosomes is seen only between homologous chromosomes. So this chromosome size differs. That is one thing. And again, in some chromosomes, out of the total chromosomes, one or two chromosomes, they are having some extra structure, but chemical composition is same. The chromosome is made up of DNA and protein. As we already said, chromatin is made up of DNA and protein. So chromatin changes into chromosome only in its morphological appearance. There is no any chemical change in the chromosome. Whatever DNA is present in the chromatin, whatever proteins are present, whatever histones and non-histones are present, all of them are same. But only thing is the chromatin, which was very long, fine and transparent and very thin, so it gets condensed. The shape only will get changed, and that forms a rod-like or a cylindrical structure called as chromosomes. So chromosomes are the condensed structures of chromatin. Then chromosomes, in some chromosomes, out of the total, take for example among the 46 chromosomes, one or two chromosomes, they are having an extra structure in the chromosome. Similar to the primary constriction, there is another constriction like this. So this structure is called as secondary constriction. So that is the primary constriction which is present in almost all chromosomes. So the primary constriction is also referred as the centromere, and the centromere is very important because it is having kinetochores. But secondary constriction is not having kinetochores. It is appearing similar to primary constriction, and it is a transparent clear zone along the length of the chromosome. And beyond the secondary constriction, the part of the chromatid which is usually small and very short, it is called as satellite. So this is referred as satellite. So satellite means it is the end of the chromatid beyond the secondary constriction. So the part of the chromatid beyond secondary constriction is called as satellite. So chromosomes which are having satellite, they are called as the sat chromosomes. So sat chromosomes means they are having satellite, and this satellite it is having some specific type of chemicals. So that is the structure of the chromosome. So usually only this part of the chromosome is taken for the study. So the kinetochores are very important. So this is the chromatid and this is the chromatid, which is also called as arm and all that. And then if you come to the next part of this chromosome, chromosomes are classified into 4 types based on the position of the centromere. Now whether the centromere is in the center or whether it is away from the center or like that. So there are 4 types of chromosomes. So based on the position of the centromere, so there are 4 types. So those 4 types of chromosomes are called as metacentric chromosomes, submetacentric chromosomes. This is another type of chromosome. Then acrocentric chromosomes. Then telocentric chromosomes. So these are the 4 types of chromosomes based on position of the centromere in the chromosome. So that means this one. Now if the chromosome, suppose here this is the chromosome. So in all cases we shall draw the diagram here. Suppose these are the chromosomes which I have drawn. So these are the chromosomes. Follow. Metacentric, submetacentric, acrocentric and telocentric. Now if the centromere is present exactly in the center, means the chromatids are equal. So this chromatid and this chromatid are equal. It is metacentric chromosome. So that means the centromere is in the center. This chromatid and this chromatid are equal in length, size. So that is called as metacentric chromosome. Now suppose if the chromatin is away from the center. So the chromatid, the centromere is away from the center. Actually this is the central region. So it is either on that side or on this side, so that the chromatids are unequal. So this is somewhat short and this is somewhat longer. So in other words, they are unequal in size. It is called as submetacentric chromosome. So metacentric chromosome, that means they are having equal chromatids, and whereas submetacentric means unequal chromatids. So one is shorter and one is longer. Now in case of acrocentric, what happens? The centromere is almost close to the telomere, so that there is one very short chromatid and one very long chromatid. See here, this is the centromere. So the centromere which is present, it is in this position. So the centromere is close to the telomere, so that a very short chromatid, a very long chromatid is seen. So that is called as acrocentric. So this metacentric means equal and submetacentric means unequal. Here also unequal, but much variation is there. So here these are of almost longer in size, but they are unequal in size. Here very short chromatid, very long chromatid, that is acrocentric. But in telocentric, the centromere is at the extreme end. That means this centromere is at this region, so that the telomere is only one end. There is only one telomere. So the centromere is extremely at the tip of the telomere, so that there is only one long chromatid. So here there is only one chromatid, and that is long chromatid. That is a telocentric. So telo means a telomere. The centromere is present at the telomere region. So acro means close to the telomere, the region near to the telomere, that is called as the acro. Meta means center, sub means away from, below the center or away from the center. So like that, based on the position of the centromere, metacentric, submetacentric. So metacentric means chromatids are equal, submetacentric means chromatids are unequal, and acrocentric means the chromatids are unequal, of course, but there is very short chromatid, and when there is a very long chromatid. So this is a short chromatid and this is a long chromatid. And in case of telocentric, the centromere occupies the telomere region, so that there is only one long chromatid, and that is called as telocentric. But acrocentric and telocentric chromosomes are very rare. But most common, we come across metacentric and submetacentric chromosomes. So whenever we observe any cell, plant or animal cell, so here the chromosomes are almost metacentric or submetacentric. So rarely, in some cases, in certain situations, we see acrocentric and telocentric. So again, among these, the telocentric is very rare than acrocentric. So overall, the first two are common type of chromosomes which are seen in a cell, and the chromosomes are they are constant in a species, and most of the chromosomes are metacentric and submetacentric. So this chromosomes which are found in pairs, they are even number, and that is about the chromosome structure, the nucleus, the chromatin, the chromosome structure, and also the position of the types of chromosomes based on position of the centromere. So this chromosome contains DNA and proteins. Chromatin also contains DNA proteins. Now this DNA and protein is always in the ratio of 50 and 50. So any chromosomes contain 50% proteins and 50% DNA. So whatever may be the size of the chromosome, whether it is a small or large, the chromosome contains DNA and protein. So what amount of DNA is present, same amount of proteins are present in the chromosome. So a chromosome contains equal amount of DNA and proteins, so that is in the ratio of 50:50. So that is the amount of DNA and proteins. So the total amount of DNA is almost equal to the amount of the proteins, which are either histones, and also there are non-histones. There are histone proteins and non-histone proteins in this nucleus or in this chromosomes. So that is about the chromosomes. And more about chromosomes, here the chromosomes which are present, they are usually in pairs. So there are 23 pairs in human being. So out of the 23 pairs, here there is one pair of chromosome which expresses the sex of the individual. So out of the, if you take the example of human being, out of the 23 pairs, one pair differs in male and female. So in male and female, so in male there is XY chromosome, in female there is XX chromosome. So that means these chromosomes are different from the remaining 22 pairs. So totally there are 23 pairs, 22 pairs, and this is the 23rd pair. So one pair only differs in male and female. The remaining 22 pairs are same in male and female. So they are not concerned with the expression of the sex of the individual. So in male, in a boy or man, the chromosomes are XY. So that expresses the male characters in that individual. In a female, girl, that means there is XX chromosomes, so that expresses the female characters in that individual. So these are called as the sex chromosomes, and they are responsible for the expression of the sex character. So that is related to the chromosomes. So that means all the chromosomes are not same. In other words, only one pair in male and female, it differs, and that is responsible for the expression of the sex character. So the baby which is to be born in a pregnant mother or in by the mother, so if the baby is having XY chromosomes, it is male. If the baby is XX chromosomes, it is female. That is how the sex is decided in the chromosome combination during the embryo formation in the mother. So that is just about some extra information about chromosomes. So chromosomes are normal, these are called as ordinary chromosomes. These are sex chromosomes. Ordinary means they are common to both male and female, hence they are called as ordinary chromosomes. Then if you come to the next part of this chapter is in the cell there are certain structures which are called as microbodies. So microbodies are also small structures which are present in the cell. For example, cytoskeleton we have studied. Cytoskeleton is a network of filamentous protein structures present freely in the cytoplasm, and this network of protein filaments in the cytoplasm is called as the cytoskeleton. It provides mechanical support, or it provides a kind of support, skeletal support to the cell cytoplasm. Similarly, here the microbodies are structures in the cell. So these microbodies, they help in the oxidation. So they are membranous structures, just like lysosomes, endoplasmic reticulum, Golgi. They are made up of membranes. Now these structures, they are concerned with the oxidation process in the cell. So microbodies are concerned with the oxidation reactions. They are usually present in the plant cells. Now in this there are two types of microbodies. Peroxisomes are one type, and glyoxysomes are one type. So these are the two types which help in the oxidation process. So this peroxisomes and glyoxysomes. So peroxisomes, they usually involve in the lipid metabolism, and glyoxysomes, they usually involve in the carbohydrate metabolism. So like that, whatever the compound on which they act and bring about oxidation, so they are classified like this. So overall, microbodies, they are associated. So they are bodies which are made up of membranes, and they are associated with the oxidation reactions or the oxidation, and they are of two types called as peroxisomes and glyoxysomes. So peroxisomes and glyoxysomes are such type of examples in microbodies. So these are the cell organelles so far what we have studied. So in the cell, diagrammatically, usually we should remember the mitochondria, the plastid, that is chloroplast, and the nucleus. So these three diagrams are important, and also the structure of the fluid mosaic model, the cell membrane. So that is also very important in this cell. So this is about the cell structure and function, where we have studied various kinds of the cell organelles in the cell. So whether it is plant cell or animal cell, So the plant cell and animal cell differs in some characters only. There is no cell wall in animal cell, but in plant cell we come across cell wall which is made up of cellulose, hemicellulose, lignin, pectin or suberin, etc. But in animal cell we don't find cell wall. That is one important difference. And vacuoles are present in plant cell, which occupy almost nearly 90% of the cytoplasmic area, so that the nucleus is shifted towards the periphery of the cell or in the cytoplasm. Whereas in animal cells we don't find vacuoles. Vacuoles are absent. If present, they are very small. That is another difference. And also another important difference in plant cell is animal cell is plastids. Plastids are present in plant cells only, whereas animal cells do not contain plastids. Plastids, they are either chloroplasts, chromoplasts or leucoplasts, which are completely absent in animal cells. Then centrosome is another structure present usually in the animal cells and absent in the plant cells. That is another difference between the plant cell and animal cell. And lysosomes are more common in animal cells and less in case of plant cells. So in plant cells they are very rare or less common, but in animal cells they are present, which help in digestion. That is the difference between the plant cell and animal cell. In the remaining characters they are one and the same. All cells are having mitochondria, Golgi complex, endoplasmic reticulum, ribosomes, then this nucleus and microbodies, all they are present in a single cell. That is the parts of the cell which we have studied in this chapter. Then in this we should remember the functions of all these cell organelles, the functions of cell membrane, the functions of mitochondria, then Golgi complex, endoplasmic reticulum, lysosomes, and also the functions of plastids, then the functions of nucleus. Functions of nucleus means it is the controlling center of the cell activities because of the DNA present in the chromatin or chromosomes. So the DNA is the molecule which expresses the characters, which expresses the activities of all the these cells in the living organisms, and that is why it is the controlling center of the cell. So this is about the cell structure and function.